It's my pleasure to talk with you today about this topic of how we've been looking at the application of nanopore sequencing as a rapid sterility measurement capability, specifically with the use case in mind of cell therapy products. So as you know, quality control, product quality control is paramount in manufacturing biologic medicines. And as we've become more and more complex in the medicines that we are making, we are looking at more and more diversity in the product and process-related impurities as we go from, large proteins like monoclonal antibodies to viral vectors and other viruses to now cell therapies. We really need more data, more opportunity to inform process control strategies, but also safety of these biological products. And so one of the major biosafety needs that applies to all biological products is the need for sterility testing. And we have certainly very good ways today to be able to do that. However, they really are not addressing the needs of the cell therapy industry at the moment. So the compendial methods, they provide a measure of whether or not there is a microbial a microbe present, but it does not identify that immediately. It takes time, usually 7 to 14 days. And the one thing that the patients who are receiving these cell therapies don't have is time. Many of them have failed multiple lines of therapy. They're trying this as a last line of therapy. They're very ill. The administration of these therapies has to be given in a coordinated manner with clinical care, where the therapy is leukapheresed, separated. There is a isolation of the T cells. There's then the introduction, the engineering of those cells to express, in this case, a chimeric antigen receptor, or CAR. The cells are, expanded, they're formulated, they're cryopreserved sometimes not, thawed and then infused back into the patient. So all this has to be coordinated. And so you really, if you have any kind of adventitious agent, you really want to be able to address that quickly. You need to use a minimal amount of the product. Those cells are very precious. You want high sensitivity and specificity, and you need to be able to detect very low numbers of microbes or fungus in this case, but potentially in the future also virus in the background of T cells, which are the actual product. So why nanopore sequencing? And boy, I need to talk faster. As you all know in this crowd, I don't need to belabor this. You have rapid species-level detection in ID. There's the opportunity for agnostic, untargeted detection. You can achieve low limits of detection. It's small, portable, and can be used at line. And some of the benefits of using a long read approach as opposed to short reads are the improved accuracy. As we know, especially now, the nanopore sequencing technology is quite high in its accuracy, simpler assembly, and the opportunity to have real-time sequence analysis. And so the work that I'm going to tell you about in just a few minutes, and you can go read the article as well, was done primarily by James Strutt, who was the lead postdoc on this work. And he set out to ask whether or not he would be able to do a metagenomic identification, broad spectrum identification of microbes that had been spiked into T cells through a combination of improvement of the experimental and the bioinformatic pipeline, and then the application of a machine learning model that could ask whether or not a sample was contaminated, and then ask whether it had been classified correctly. And so the sequencing workflow is very standard, as you see here, with the exception that he was taking advantage of the highly conserved regions of prokaryotic and eukaryotic genomes that allow untargeted approaches for this sequencing. And so he would start by spiking these into the T cells, and then extracting the DNA and going through all the different steps, experimental steps of the pipeline for doing ONT nanopore sequencing. Then he implemented a bioinformatics workflow that, in addition to all the things that we do, so the base calling and the read trimming, he was also implementing a host removal step that bioinformatically removes some of the host reads, and then undergoes metagenomic classification, and then to identify these contaminant candidates that then go through the machine learning algorithm and essentially a confusion matrix or a decision matrix that allows you to assign them. So what he was able to show was that he is in fact able to detect both bacteria and fungus down to 10 colony forming units per ml, which is what is required by the regulators. And he was able to do this in about 16 hours. And he was able to detect aerobic and anaerobic species, difficult to grow bacteria, and so forth. And then he was able to show that the model he created was also, in fact, able to detect and appropriately classify the contaminated samples as contaminated and sterile samples as sterile in 32 out of 32 cases or 31 out of 31. Where you see we still need to do more work, and we're working on this today, is in the negative controls, where we are only predicting those correctly 18 out of 24 times. And of course, there are a lot of reasons why that might be. The amount of data you include in your training data. We have some strategies around how we're approaching that today that we can talk about maybe in the discussion as is appropriate. So in the next 3 minutes, I just want to touch on the question of what about viruses? Why shouldn't we be testing for adventitious viruses in cell therapy products? It's not required today. And many cell therapy processes remain manual and open. Human cells are more susceptible to replication of a variety of human viruses than our CHO cells. We still use a lot of animal and human-derived raw materials when we're making cell therapies and other, ATMPs. And of course, there are no downstream viral clearance steps that are possible for cell therapies because you would lose your product if you did that. So we think nanopore long-read sequencing actually could provide in the future a rapid assessment of viral pathogens during cell therapy manufacture. I know that this has been the topic of many other sessions here today in terms of the application to many different types of biologics, but in particular for cells, I think it also has applicability. And some of you may have seen Dr. Charlie Swafford's talk this afternoon. I don't know if some of you saw his talk, but if you didn't, because there were concurrent sessions going on, what you would know is that one of the main challenges is around the fact that you have the presence of host cell genomic DNA that obscures your ability of detecting the virus. It's essentially like looking for a needle in the haystack when you consider the length of the host cell genomic information compared to the viral or bacterial information. And so what Charlie and Emmanuel shown here, and to some also James helped a little bit on the bioinformatics, what they decided to do was to approach this, and others are doing this too, through host nucleic acid depletion and viral nucleic acid target enrichment. And so just briefly, because I don't really have time to go into the details, what they did is they developed a new workflow. So you see the standard workflow that's shown here, where they've spiked MVM into cells, and they have gone through the standard workflow. They created a new workflow that they call ConSeq, where it's concentration nuclease treatment, DNA, RNA, and extraction, followed by an agnostic PCR step called CISPA. And they've replaced those two steps that are shown in red there with these. And in doing so, they've been able to really reduce the limit of detection by 3 log. And they've shown that for 3 different types of viruses, but I'll just show you this one set of data here in the interest of time. So in summary, I hope in 10 minutes I have convinced you that amplicon-based nanopore long-read sequencing enables agnostic detection of microbial contaminants down to 10 CFU in donor T cells, that the ConSeq pipeline that I showed you can enable rapid detection of viral contaminants in JURCAT T cells down to 1 vector genome per 1,000 cells, and that nanopore sequencing has biosafety applications in biopharmaceutical manufacturing today for sterility testing, I believe, and also for virus testing. It really has great potential to be used in these contexts, and I think you'll see more as the technology continues to advance. Thank you very much for your time. I'd like to acknowledge all the researchers and collaborators that worked on this with me. Thank you.